So again, my name is Mary Krauszer, and I am here to talk to you about my research on sperm longevity under different short-term storage conditions. So as Erin mentioned, my species of interest for this project is the pinto abalone, which is a large marine snail whose range extends across the coast of North America from Alaska down to Mexico. And you might recognize abalone shells, which are commonly used in jewelry and other adornments, or maybe you have eaten abalone, probably not this particular species. There are over 50 species of abalone around the globe, and everywhere they occur, they are appreciated for their beautiful shells and as a food. Here in Washington, the pinto abalone is our only abalone species, and they have cultural significance to many Coast Salish tribes. And as you can see from the diver in the background of this photo, our pinto abalone live deep in the water on rocky substrates, and they're often associated with kelp beds. And as grazers, they play a role in cleaning off rocks for the succession of macroalgae like kelp to be able to take hold. There was a sport fishery for abalone here in Washington that began in the 1950s, but unfortunately that recreational harvest as well as illegal harvest really impacted wild populations, and ultimately the fishery was closed by the mid-1990s. But even with that primary pressure of fishing removed, abalone populations did not recover but continued to decline. A early 2000s study by the Puget Sound Restoration Fund detected no young or juvenile abalone recruiting to wild populations, indicating that reproduction is failing in the wild for this species. So recognizing their cultural importance, their ecological role, and those observed population declines, the species was ultimately listed on the state endangered species list in 2019. And unfortunately, it makes sense that reproduction is failing for this species in the wild because of the particular reproductive mode of these snails. Abalone have separate male and female sexes, but rather than mating through copulation, they broadcast spawn, they release their sperm and eggs into the water column. And so if you have really low numbers of individuals and therefore large distances between individuals, it's much harder for sperm and eggs to meet in the water column to produce larvae. And for that reason, recovery efforts for this species here in Washington have focused on captively breeding abalone in hatcheries for release into wild populations to increase both the number and the density of individuals. So as you can see here, abalone are produced in a hatchery, marked with a little ID tag, and then released out into those kelp beds by divers. And one of the primary hatcheries doing this work here in Washington is the NOAA Manchester Chew Center for Shellfish Research and Restoration, operated by the Puget Sound Restoration Fund, with Josh Balma and his team pictured there, who I had the pleasure of working with on this project. So to produce abalone in the hatchery, staff separate the snails into their individual buckets, expose the animals to a chemical bath to induce spawning, and then collect eggs and sperm and manually mix them to make the desired crosses to produce larvae. But executing this spawning protocol in the hatchery is quite labor intensive and time intensive. It's done entirely in the dark, which is why Jen is checking on those abalone with a red flashlight. And the animals don't always respond reliably to the spawning protocol. Some individuals might spawn right away, others 6 or 8 hours later. Some don't spawn at all or only a little bit. So there can be timing mismatches between when eggs and sperm from the individuals that you want to cross are actually available. And this is further complicated by the perishability of eggs, which only last about 2 hours. So you really need the timing of eggs and sperm and their availability to match up. Under current hatchery protocols, only fresh eggs and sperm are used for these crosses, and sperm is generally disposed of after 8 hours. So to address this timing and availability mismatch between sperm and eggs, I set out to investigate sperm storage techniques. Previous work by Caitlin O'Brien at the Puget Sound Restoration Fund focused on long-term sperm storage through cryopreservation, but my study honed in on short-term storage techniques. So I focused on understanding whether we can store sperm and keep it usable for longer, with the goal of having a ready supply of sperm at the hatchery for days rather than hours. I investigated short-term storage conditions that are common in other aquaculture applications, which are temperature, sperm concentration, and calcium reduction in the storage media by diluting the sperm into a saltwater solution without calcium. And each of these storage conditions works by affecting sperm activity or activation. So as we probably all know, sperm swim with their little tails or their flagella, but they only have so much energy in their cell to power that swimming. And prolonged swimming can trigger cell degeneration in other ways. So the idea of these storage conditions is to delay or depress that swimming activity while in storage, so the sperm can be activated later for use. And how well a sperm is still swimming can give us an indication of its health. So sperm motility is often used as a proxy for sperm viability. So my measured response variable in this study was that percent motility, or the proportion of sperm in a sample that are still swimming like healthy normal sperm. And my research questions focused on whether I could apply these short-term storage conditions to pinto abalone sperm specifically and see the motility remain high for longer than 8 hours, which is again that kind of current hatchery shelf life of sperm, and to understand what effect each of these storage conditions had on motility longevity. To answer these questions, I collected sperm from 8 male abalone and subjected samples to the different combinations of storage conditions. I then observed sperm activity by recording recording videos, with a high-speed microscope-mounted camera. First on the day the sperm was collected, and then every day for 6 days after. And before observing the sperm, I would take it out of storage, activate it by diluting it in seawater and warming it up to hatchery temperature, because again, the idea of the storage technique is to dampen activity while in storage and then activate the sperm when you are ready to use it. And for my work, I followed the methods from many previous studies, but I had to figure out how to do this at Evergreen with the equipment available to me and or that I could purchase. So before I got my hands on abalone sperm, I spent several months last summer figuring out how to test out different combinations of microscopes and cameras and software and microscopy modes, all using my oyster sperm as a model to prepare for the abalone sperm. And my equipment setup was a little complicated because since we know temperature affects sperm, I had to make all of my observations in a temperature controlled environment. So that picture on the bottom right is my little walk-in refrigerator where I made my observations with my mini fridge inside of it. So lots of time in the basement last summer when it was very warm outside. But once I was ready for the abalone sperm to do my real data collection, I had the pleasure of partnering with the Puget Sound Restoration Fund and the Chu Center to spawn abalone up in Manchester and then transport it here to Evergreen to assess motility. And since I was limited by the type of microscope that I was allowed to take into the cold room with me, I captured videos under bright field microscopy, which produced the raw video you see here, kind of grainy black cells on a gray background. I then processed the videos with an open source software called ImageJ, and with a custom macro that I wrote and some manual processing to produce binary files of white cells on a black background. And I was then able to analyze the videos using computer-assisted semen analysis, or CASA, in a system called OpenCASA. And CASA systems were developed for use originally in human fertility, of course, but have been adapted for use on many other species sperm by adapting the parameters such as speed and sperm path that are used to identify a motile sperm. So I developed some parameters for pinto abalone sperm based on previous work by Caitlin O'Brien and then some of my own calibration. And ultimately, the CASA analysis yielded a percent motility value for each video observation. So moving on to results. I'll show just a couple of the figures from my work that I think illustrate my key results and findings, starting with a summary figure. So on the y-axis here, we have our percent motility or percent motile sperm. And on the x-axis, we have the days since the sperm was collected. So day 0 was the day of collection, day 1, 1 day after, and so on. And each data point represents a sample from an individual abalone, and then the mean is represented by an asterisk. So in our first gray panel, which is less gray on the screen, I guess, we're looking at the samples that were stored diluted in seawater and at normal hatchery temperature. And you'll notice that motility drops to 0 across all samples by day 3, and it's a pretty uniform drop-off for these sperm samples that were stored at what would be kind of normal hatchery conditions. In the next panel, we have the samples that were refrigerated and stored in seawater, and you can see there's a much larger spread in motility values across individuals, with some samples remaining at pretty high % motility into days 4 and 5. And the longest lasting samples here are those with the highest sperm concentration. And similarly, the samples that were stored refrigerated, but in the calcium reduced media, have a larger spread as time goes on. But again, those that remained at pretty high motility were those with the highest concentration in storage. And these observations showed that at least some samples remained over 85% motile up to 72 hours after collection under refrigeration and high concentration. And then beyond just describing the longevity I observed, I also used mixed effects modeling to assess the effect of each of the storage conditions on motility. The fixed effects I included in my model were the 3 storage conditions of temperature, concentration, and dilution media, as well as the age of the sperm in hours and an age squared term. And then individual was included as a random effect to account for variation between individual abalone and the fact that I was doing repeated measures over time. And in all, the model had a marginal R² value of 0.75, meaning that 75% of the variation in motility was explained by the variables considered in the model, so pretty strong explanatory power. And the figure here is an effects plot showing just one of the variables in the model, concentration. So the effects plot shows the predicted values from the model with concentration as the main effect. So we have, again, % motility on the y-axis and then the log of concentration on the x-axis. So this visual is showing us that when accounting for all other effects, motility at a given time is higher when concentration is higher. And the predicted values here are color-coded, so you can see that the effect of concentration on extending motility longevity is stronger under refrigeration, which is the kind of blue color data points. So ultimately, I was able to answer my research questions in that, yes, I could keep sperm over 85% motile for a few days, which is definitely longer than 8 hours. Refrigeration resulted in higher motility rates over time compared to hatchery temperature. Storing sperm more concentrated had a significant extending effect on motility longevity, and the effect was stronger under refrigeration. And finally, diluting sperm in a calcium-reduced media for storage had a borderline non-significant but positive effect on motility. So with this project, I set out to investigate short-term storage techniques to extend the motility longevity of pinto abalone sperm for use in captive breeding programs. And my observations demonstrated that sperm can be stored for days at a time and remain at high levels of motility, with the combination of refrigeration and storing sperm highly concentrated proving the most promising for extending the shelf life of sperm. It is important to note that my study used motility as a proxy for viability, but fertilization is really the gold standard for doing that. So future studies should focus on performing fertilization trials with stored sperm. And applying these short-term storage techniques may allow hatcheries to maintain a ready supply of sperm for days at a time instead of relying solely on fresh sperm. And extending the shelf life of sperm could increase hatchery managers' control of genetic crosses in the captively bred abalone. And stored sperm could also be shared across facilities or even to mobile spawning units to expand the efficient use of gametes in this endangered species, with the hope of producing juveniles that can be released into the wild to increase the density of wild populations to a level where they can reproduce successfully on their own and achieve recovery in Washington state. This study was made possible by funding support from the Washington Department of Fish and Wildlife's Shoemaker Internship Program and by significant staff support and gametes from the Puget Sound Restoration Fund. I would also like to thank Drs. Pauline Yu, John Withy, and Erin Martin for their guidance on this project. And with that, I will open it up for questions.